Hello biologists, this is Miss R. We're studying for quiz 1.12 and quiz 1.14. We're talking about how RNA makes protein. That's translation. Protein's important for a lot of things in your body, including your beautiful hair, whether you're a polar bear, a fox, or a human. If you want to make beautiful keratin for your hair, the cells in your scalp make that protein. And in order to make that protein, they need to do translation. We've talked about how DNA makes DNA, that happens in the nucleus. We've talked about how DNA makes RNA, how the big book of how to cook everything, which is your DNA, gets transcribed into a smaller, single-stranded recipe card that can travel outside the nucleus. So transcription, this process of creating the recipe card, happens inside the nucleus, but then that recipe card travels outside the nucleus to make protein. So there's kind of a nucleus boundary right here between B and C. Translation outside the nucleus, mRNA travels from the nucleus, works with ribosomal RNA and transfer RNA to make proteins. How do you make a protein? Well, we need to think about where it happens, what tools you need, and what ingredients are necessary. Just like every recipe needs ingredients, a protein needs ingredients as well. So let's go back to transcription for a moment. We take that big book of how to cook everything, all of your genes, the codes for everything are contained in your DNA in the nucleus. If you want just one recipe, one gene, one protein, you transcribe just one gene at a time, or your cells do that for you, and you create a single-stranded piece of RNA called messenger RNA. That's what it ends up, and that travels outside the nucleus into the cytoplasm where the ribosomes are. Here's another view of it. Here's the messenger RNA, and here's it traveling outside to the cytoplasm. This is messenger RNA as well. It's a message from the nucleus, from the DNA, to the ribosomes. Here's how to make a protein. So think of that M as M for messenger. Messenger RNA is arranged so that 3 bases code for 1 amino acid, and a string of amino acids make a protein. The tRNA reads this code on the mRNA. So the codon is here on the mRNA. It's a group of 3 bases, and the tRNA reads the code on the mRNA and brings in an amino acid. So the messenger RNA you can see is right here in the green. The amino acids are right here attached to the tRNA, which is set up so that it has the opposite bases of the messenger RNA. And this is all happening at the ribosome. This big blue glob is the ribosome. Codons on the messenger RNA match up with anticodons, this piece here, that matches up with the mRNA on the tRNA. tRNA has the anticodon, mRNA has the codon. So the codons here are in the mRNA, the anticodons are in the tRNA. Remember, the real recipe is in the mRNA, and the tRNA is just reading. It has the anticodon. It can decode. So here's this sort of 3 different views of transfer RNA. The anticodon is at one end, and this end is where the amino acid is brought in. And there are different types of tRNA for all the 20 different amino acids. It kind of works like a grocery cart. You go to the grocery store with your recipe to get ingredients, and the mRNA comes to the ribosome to shop for ingredients, and the tRNA is like the shopping cart. It runs out and gets the amino acids that you need for your mRNA recipe. Ribosomes are where this all happens. They're located outside the nucleus. Some of them are floating around in the cytoplasm, and some are located at the rough endoplasmic reticulum, which is kind of around the nucleus but not inside it. The small part of the ribosome usually attaches to the mRNA first. Then the larger part attaches, and the tRNA, represented right here, comes in. Here's a nice animation of the translation process. You can see the mRNA is coming outside the nucleus. It's lining up there. It's got the UUU as a signal to start. Then the transfer RNA is right here in the green, is reading the code in the mRNA. That's where the codons are. The ribosome is dissolving away here. You can see there are multiple ribosomes working on one mRNA. There's the protein, and it's folding up. Here's another nice view. All right, we have to transcribe first. It's reminding us that we need to transcribe before we can translate. So you can see that the DNA is being unzipped here. This is transcription. You can tell it's transcription and not replication because only one side of the DNA is being used. That's the sense side of the DNA. The antisense side of the DNA is left alone. The stop codon appears. And now we need to translate. Okay, now we can translate. So remember, the mRNA leaves the nucleus and heads out to the rest of the cell where it finds the ribosome, large and small subunit. You've got your tRNAs coming in. They're bringing in the amino acids. And this model is especially nice because the amino acids start to form a polypeptide chain, and you can see how the amino acids interact. The charges on the amino acids make it form a particular shape. And so the protein starts to take on its characteristic shape. And each protein that your body makes has a characteristic shape. And if it doesn't assume that shape, it doesn't work. Remember, it's the transfer RNA that's bringing in those amino acids. And this is all happening outside the nucleus at the ribosome. Here's where we did transcription, inside the nucleus, and that mRNA traveled outside the nucleus to the cytoplasm where the ribosomes are. The tRNA is working with the ribosomes to bring in amino acids to make that protein chain. Can you label this diagram and explain what's happening? See if you can put in codon, anticodon, tRNA, mRNA, rRNA, that's ribosomal RNA in case you forgot, polypeptide, and amino acids. Pause the video and see if you can label everything. Okay, let's see how you did. If you labeled this part, the protein chain, this is a better one because it has different amino acids. That's the polypeptide, or the forming protein. Okay, so we have polypeptide, we have tRNA. This green glob here is the the rRNA, the ribosome. Right here, that's a codon on mRNA. This, of course, is the mRNA with the codon, and the anticodon is on the tRNA. That's the other side. And this curvy deal, that's the tRNA. So let's test your knowledge a little bit more. Get you ready for the quiz. So can you tell what amino acids will be? How can you tell what amino acids will be in a protein? You look it up in the codon table, you look at the gene. The mRNA has codons for specific amino acids. The DNA codes for the amino acid sequence, or E, all of the above. Take a second to think about it. I'm going to show you how the amino acids that have charges are attracted to each other and start shaping proteins. So positive and negative attract, and you get proteins that start to curl up and have shapes, and it's the shapes of these proteins that make them unique. If you guessed E, all of the above, you're absolutely correct. You can look at a codon table to figure out what the mRNA says. You can look at the gene in the DNA, because the DNA, of course, codes for the amino acid sequence. It codes for the mRNA that has codons for specific amino acids. The anticodon, remember, is here on the tRNA. Codon on the mRNA, anticodon on the tRNA. How do the amino acids arrive at the ribosome? How do you get those different parts, those pieces of a protein? My protein beads here have lots of different amino acids in them. They arrive by taxi, plain, tRNA, mRNA, or double-stranded RNA. If you guessed transfer RNA, or taxi RNA, as some of my students like to refer to it, you are correct. Where are the instructions to make a protein? Are they in the tRNA, the mRNA, or the double-stranded RNA? Take a second to think. I'll give you a hint. It's the messenger RNA. The genetic code is right here. It took us scientists a while to figure this out, but now we know if you have G, A, G, that would be glutamine. And isoleucine, LIE, which is right here, can be coded by 3 different codons, AUU, AUC, or AUA. Remember, amino acids are the parts that you link together to make the protein. And proteins are what make up you. Hair, bones, teeth, skin, eyes. You're made of protein. Where does the original information come from to make a protein? Is it inside the nucleus, from DNA, in a gene, on a chromosome, or all of the above? If you guessed all of the above, you are absolutely correct. Before we go, let's talk about what proteins are good for anyways. They're enzymes. They catalyze chemical reactions like replication, transcription, and translation. They're good for transporting things within your body. Hemoglobin binds oxygen and transports it within your body. Holds reserves of elements and compounds. That's storage proteins. Contractile proteins are in your muscles. Structural proteins like keratin and collagen make up you. Your antibodies are made of protein, and there are regulatory proteins like insulin that regulate your metabolism. So now you know how to get beautiful hair. Your scalp cells make it, and they do it right here in the ribosomes. Ribosomes, of course, speak many languages. This is an interesting diagram of Japanese. See if you can explain it to yourself. Pause the video, give yourself a minute or two to try to name all the parts and explain how it works to yourself. If you guessed this is the ribosome, this is the tRNA,